All right, my friends, welcome. Welcome. It's nice to see so many of you here. It would be even nicer to see more of you in the Count 2 room in the back channel. If you're into that, it'll be helpful. You've taken the effort. You've taken the effort to come to class. So if you take that little extra effort, you'll even get more out of this class. It has always been sort of my dream to have much more active learning in large classes. The data are really clear that students tend to learn more if they're more active. Your chance of failing the course goes down 20%. In courses that are much more active. But we have this huge room and lots of people. So anyway, we're going to try not to let that stop us. I have a camera today just to record what goes on for my own purposes. I don't want you to freak out. I'm not going to send it to your parents or show it to people. We're finishing up cycle 7. Why is it that whenever there's a camera, people have to wave at it? I don't know. It's a human thing, I guess. We're finishing up cycle 7 today and moving into 8, and it's all about gene expression. Building on things that you learned in high school and taking them a little further. Just a heads up, module 4 has substantial homework, and you need to be in communication with your team. So get together with them to talk about that. We're going to have a GSA this weekend, and then the weekend after that is another term test. So do this. I'll help you all along the way, but again, you have to help yourself. Make these lists, draw pictures of genes, show the pictures to your friends. You need to draw on your friends. Many of you, well, you all have varied backgrounds from high school, so you might find that you know something your friends don't, and vice versa. So your study groups will be really, really important moving into this term test. So before we meet with this class, I often ask you what information is still bugging you, what's still troublesome, and this is the response from the last time, from today. Again, I'm trying to help you with this. So when you just put bullshit in there, it doesn't help me. When you say, oh, I don't understand why Maxwell is so cool, it's funny, but it makes my job harder, sorting through things that aren't relevant. So let's take this seriously and let me know what's having trouble. So SD boxes, operons, UTRs, downstream and upstream. So I've tuned this class a little bit, as much as I can, to these things. We're going to talk a lot about operons. And the SD box, I'll try to figure out how to address that particular issue. But again, you need to also help yourself. Talk to your friends. Come to office hours. Go to the mentor workshop this week, Thursdays, I believe they are. Like, get out there. Don't let this stuff slide. I have a clicker question coming. Count 2 poll coming. Get ready. But before we do that, I want to make little clusters like we did way back in the first term. Okay, so people along this wall are number 1. People along this edge are number 1, and just count off. 1, 2, 3. Make little groups of 3. People on this edge are 1. Just count off. Hold up your finger. 1. Are you 1 or 2 or 3? Count along. Count along. Just going to make a little team. Okay. Make a little team. Shh. I know some of you hate this. I hate it. I'm an introvert. I don't like talking to people I don't know all the time, but it's really, really, really helpful. So even if you are kind of introverted and you don't like the smell of the person beside you, talk to them about this. Which of those ribosomes, A, B, C, or D, has been translating the longest? Talk to your new friends about this. Which of these ribosomes is the oldest? Which one has been translating the longest? And shoot your answers into the count to poll. Which of these ribosomes is the oldest? Which one is the oldest? Okay. Votes are in, and we're arguing over A and C. Remember we said in the last class, multiple polymerases can transcribe a gene at the same time, and multiple ribosomes can translate a message at the same time. So polymerases start here on the right, and then they go this way. So this is the oldest polymerase. The one on the left, your left, is the one that has been transcribing the longest. That's the oldest polymerase. As soon as a little bit of message comes out of a polymerase, a ribosome can jump on there. As soon as the first bit of message comes out of a polymerase, the SD box is right there, and ribosomes will jump on that and start to translate. And they'll translate right along behind the polymerase. So yes, which one is it? A. This ribosome, number A, has been translated all the way from the end all the way up there. Ribosome number A has been translating the longest. The ribosomes are moving up this picture, up the picture. The polymerases are going right to left. So this is a really, really important diagnostic image. If you get this, if you can get the movement of this, then you are in good shape. If this is still bugging you, then again, argue with your friends. Argue with your friends. Now we need to talk about operons. Operons are really cool because in bacteria, sometimes, if you need one gene product, you need a bunch. If you need one, you might need 3 or 4. So why not put 3 or 4 genes under the same control? Why not put 3 or 4 genes under the control of the same promoter? Cool. That's all an operon is. It's just several genes under the control of one promoter, instead of each gene having its own. Instead of each gene having its own promoter, they have only one. But that means something amazing happens. That means the message is coding for many genes. The message codes for multiple genes. Not just one, like you're used to. So there's going to be a start codon at the beginning of this, and at the end of each gene, there'll be a stop codon. And then another start, and then a stop, and a start, and a stop for all of those genes along the way. Because the start and the stops are so close, because they're so close, the ribosomes never really get away, and therefore we do not need SD boxes. We need only one SD box here at the start, at the beginning, to get the ribosomes on here, but then we do not need SD boxes for the subsequent genes. The ribosomes are just right there anyway. They've just finished translating the previous gene, and they jump right on the next start codon easily to continue their translation. Okay with that? I realize SD boxes are troublesome for some folks. Please don't hesitate to put your hand up or shoot a question onto the back channel. Okay, but if you've got all these genes under the control of one promoter, well, then you could regulate that. You could regulate the activity of this promoter with another sequence called an operator. The operator is, again, a protein binding domain. It's a place that proteins can bind. It's understood as double-stranded DNA. And in the case of the trip operon, this operon is making the biosynthetic enzymes needed to manufacture tryptophan. So again, if you need one of them, you need them all. You need the whole pathway. But tryptophan is an amino acid, and if we can get it from the environment, we don't need to make it. We don't need to express all of these genes. So that's what this blue thing is, a repressor. A repressor that can bind onto the operator region. It's a DNA binding protein. And this protein is coded by a gene called TRPR for regulatory or repressor. So TRPR is an independent gene, has its own promoter, its own SD box, its own start and stop, its own transcription terminator, all of those things. It makes a protein, the TRP repressor, that can bind onto the operator. If TRP repressor binds the operator, then it blocks transcription. Okay, so it's really no more complicated than that. A bunch of genes under the control of one promoter that is in turn regulated by the binding of a repressor onto the operator, and the repressor is coded by an independent gene, TRPR. Now, of course, what makes this cool is that this system can sense the presence of tryptophan. If there is already lots of TRP, then here, the green thing is TRP. If there's lots of TRP in the environment, it will bind onto the repressor, and then the repressor can bind the operator. Tryptophan binds the repressor, and that enables the repressor to bind the operator. And that dramatically reduces transcription, dramatically. I don't want you to think of any of this regulation as an on-off switch. Not on-off. They're just dimmer switches. The repressor binds to the DNA, but then it comes off. Binds again, comes off, binds, comes off, binds, comes off. And when it's off, a polymerase could sneak through. So they're really just dimmer switches. Nothing really is off in these cases. Now look at this. On the left is the TRP repressor protein. There's a pink one and a blue one, just to show you that it works as a dimer. A single TRP repressor doesn't work. It has to pair with another one. It works as a dimer, and in that form it's called this aporepressor. It doesn't actually work in this form. This dimer doesn't work yet. But when the dimer binds to trip, see this? 2 molecules of trip. You can see how it changes the shape slightly. Changes the shape slightly. And then the repressor can bind onto the operator region. The repressor is non-functional when it's made, but in the presence of trip, the repressor binds the operator and shuts off transcription. Any questions about that? This is a biosynthetic operon. If we can get trip from the environment, or we've already made enough, then that excess trip will bind onto this repressor, allowing it to shut off transcription. Yes?
Is the regulatory gene in close proximity to other genes?
The regulatory gene for trip is not really close by. It's not really close by, but it doesn't need to be. Good. Good question. Something else? All right. Excellent. Okay, so that's the basic system. If trip is available, it binds the repressor and shuts off transcription. So now let's think like geneticists. This is the drawing part. Let's look at some axes here. And again, you'll get more out of this if you do the drawing, at least talk about it in your group. So this is expression of the trip operon. Trip operon expression. And this is just over time. So usually the expression of the trip operon is pretty high, right? I mean, these genes their default option is on. We are getting expression because we need to make trip. But right here we add some trip into the solution, into the growth medium. We add some trip. And what you would expect, of course, is for the trip expression to fall off. It's not going to go straight down, but it's going to fall off. And it won't go to 0. The literature says it comes down to about 1/70th of its original. So this is normal. This is wild type. This is what the phenotype is for a repressible system. It's being repressed by trip. Now, this is all good, but what I'd like you to do is think about a mutant. So we've got a mutant. A mutant, it expresses like usual, like usual, like usual, like usual. Add the trip, like usual, like usual, like usual, like usual. So this is a mutant. I want you just to think. Don't talk to your friends, just think. Where might that mutation be for a strain that would behave like this? Just think. Where might a mutation be that would cause a strain to react like this instead of normal, instead of wild type? Just think. People are talking. Shh, don't talk. Just think. Just think. Get your idea. Okay. Your first idea probably is too obvious or easy. Pitch it out. Throw out your first idea. Think of another one. What's another way, another place a mutation could be to explain this phenotype? Another place. Okay. Now just tell each other in your little group of 3 where your mutants were. Compare. See if you have the same idea. Okay, let's get some ideas here. Here's the operon, here's the behavior of a mutant. Let's take a couple of ideas. Where might the mutation be? Where might it be? Let's start more kind of near the back. That's as far back as we can get. Shout. Maybe the mutation is in the operator region, such that it is now not attractive to the repressor at all. The repressor is not binding to the operator. We've lost the operator or we've mutated it in some way. Lovely. Another one. Let's come down. Yes, sir. The gene that codes for the repressor protein has been inactivated. In what way? Anybody? What might the mutation be in the repressor gene? Where might it be? Where might it be? Yep, follow up. So maybe it's in the tryptophan binding site, the mutation in the tryptophan binding site. You had another idea?
Also, when so it can't bind to the dimer.
Maybe it can't make a dimer. Maybe it can bind TRP, but it can't bind another repressor, so it can't make a dimer. Maybe the mutation is in the promoter of TRPR, so the promoter doesn't work. So TRPR isn't even transcribed. You see, there are many, many places that mutations could be to give this phenotype. I would invite you to play this game with your study groups, with your friends. Make a mutant and challenge them to figure out where the mutation is. Show them a phenotype. Show them a phenotype and challenge them to figure out where the mutations are, or vice versa. Okay, these are terrific, terrific test questions to help us understand better. Okay, so making mutants and studying how mutants behave is a great way to study this stuff with your friends. Okay, I'm going to change gears here now for a moment. Tell you this little story about this guy named Mr. Maxwell. Mr. Maxwell used to be the assistant dean in the Faculty of Science, but that's a very stressful job. So he decided to quit all that and move to Northern Ontario and live off the grid. Quit all that, live a stress-free life off the grid. So he built himself a house and a garden, and he dug this stream out of the river to provide water for his garden and for drinking, but also for power. So he's powering his operation with a water wheel generator, and he's watering his crops. So many of you have the beginning of this image if you've downloaded the slides. If you haven't, then you can see on your monitor, and I will hand out some in a minute. Because Maxwell was so happy with this life that he started bragging to his friends about how cool it was, so his friends showed up too. So his friend shows up, and they build a house and a garden, and they want some water too. They want some water. So you've done these exercises before, these invention exercises. So I will show you this picture so you can make a sketch if you need to, but I want to show you the challenge. So in your group of 3, your team your team needs to come up with an invention to regulate the flow of water to these 3 places. Regulate the flow of water to these 3 places. Okay, let's get ready. Reminding you that we have this river. We're pulling water from the river. The river never runs dry. We're pulling water from the river into the stream and then into these little branches that are powering each of these 3 houses. The water requirement for these 3 houses is variable, goes up and down. So we're trying to invent a way to make sure that everybody gets the amount of water that they need when they need it, and not too little and not too much. Is there an inventing team who would like to come and show their solution to this problem? Is there anyone who would like to come up and demonstrate how cool their invention is? Yes? Excellent, thank you. Okay, shh. What's your name?
I'm Caitlin.
Caitlin, okay.
So I don't know if it counted as closing the gates or not, but so we put gates in by all the houses. So in times of really high water, we figured this gate could close like halfway, so that way not a ton of water was getting through. Or in times of low water, it could open up all the way, or to conserve water, close a bit. And the same at all the houses, if there was lots of water in this stream area, to close to make sure they're not flooding, but then open up if they need lots of water.
Nice. Okay, so 2 sets of gates, one at the major opening of the stream, and then smaller gates at each of the properties. Okay, nice. Can we keep this here for now?
Sure.
Great, thanks, Kate. Does anyone have a different idea, a different invention, a different way to solve this thing? They do? They don't want to show it. Yes, come on up. Good. Hey, what's your name?
Okay, so basically we have 3 separate reserves.
Yeah.
Okay.
That's good.
Okay, for each of the houses, that will always be at the same volume based on pressure and volume differences. And they can basically have access to water any amount that they want from the reserve, and you can basically pump water from the stream to the reserve by the generator.
Excellent. Okay, can I keep this here for now? Okay, great, thank you. Okay, so we really only have time to do, what am I speaking to this for? We have only time to do these 2, but you'll notice they have the same characteristics. That is, they have local control on each individual homestead, local control, in addition to some kind of global control. So that's what this is. The repressor binding is global control of transcription, right here. The repressor binding is a global control. Remember, it's not an on-off switch. It's just a dimmer where we can drop expression down to 1/70th. But what's just so amazingly cool about TRIP is that it has another level of control. It has local control as well. Local control as well. And that's what I want to try to help you understand. This is not in the textbook. Focus right here on the operator. Right here on the operator. I'm going to change the image. The graphic style will change, but we're focused on the operator. Okay, same idea. Look, promoter, operator, but right here, right beside the operator, is another freaking gene called TRIP-L. Its product has no function. This is so cool. Its product has no function except to get made, except to get translated. And that sounds really crazy. A new gene called TRIP-L is coded right here in the 5', what would be called UTR, a new gene whose product has no function other than to get created. So now here's a tricky leap. Remember, in the previous class we talked about those hairpin loops? So you already know that mRNA can pair with itself. Put that on your list of complementary base pairing. mRNA can pair with itself. So look at this. There's this region called the attenuator. Attenuator. Attenuation means sort of dimming. Attenuation means fine detail changes. The TRIP attenuator, once it's transcribed, once that region is transcribed, look, it can pair with itself. This is the local control. This is the control that's right at Dennis's house. The TRIP attenuator, after transcription, can pair with itself. And look at those 4 regions. 4 regions. I can see 3 people who seem to be sleeping. Could you just kind of give them a nudge, because it would be a shame if they missed this part. I think they fell asleep during the activity, but now if they could catch this, it's important. Look, the TRIP attenuator region as mRNA, it's understood as mRNA. It can make these hairpin loops. Keep your eye on this loop, the 3-4 loop. The 3-4 loop is a transcription terminator. And you might be thinking, bless you, you might be thinking, oh, that's weird, Tom, because transcription terminators are supposed to be at the end of genes. Yeah, that's the cool part. The TRIP operon uses a transcription terminator at the beginning as well, and I'll show you what it's doing. So look, I'm going to show you this a couple of different ways. These orange parts are TRIP codons. Those orange parts are calling for TRIP tRNA. So when a ribosome begins to translate, it's going to ask for TRIP tRNA. And look at this. If the ribosome gets TRIP tRNA, that must mean the cell doesn't need to make TRIP. If there's TRIP-charged tRNA available, that means there's lots of tryptophan. The cell does not have to make TRIP. And look, the ribosome proceeds beyond those trip codons and blocks the formation of the 1,2 loop, that allows the 3,4 loop to form, and transcription stops. That 3,4 loop is a terminator. Transcription stops. If tryptophan is available, the 3,4 loop forms and transcription stops. If trip levels are very low, trip-charged tRNAs would be very rare. And a ribosome asking for one of those trip tRNAs wouldn't get one. So the ribosome will stall. This is so elegant. If the ribosome stalls here, 2 cannot pair with 1, so it will pair with 3. And that will prevent 3 from pairing with 4. That will prevent transcription termination, and transcription will continue, as we would hope. If the ribosome cannot find trip-charged tRNA, then we want the operon to be expressed. And we ensure that it is expressed by preventing the formation of this terminator loop. The blue structure is the ribosome translating this message. Good question. Thank you. The blue structure is a ribosome translating the message.
What do you mean by trip-charged tRNA?
Excellent. tRNAs bring amino acids to the ribosome. Is that you? Who has? tRNAs bring amino acids to the ribosome. tRNAs, when they're first made, they don't have an amino acid stuck on them yet. Enzymes attach that amino acid to the tRNA, and that makes it charged, trip-charged tRNA. Nice, thank you. Please, yes.
This green strand is mRNA complex.
The green strand in this picture is mRNA. mRNA pairing with itself, just like it did in our previous class, to make a transcription terminator loop. Let me show you this one more way. One more way, and then we'll. Okay, one more way. Watch this and be careful. The bottom of this picture is the DNA. You're used to seeing it the other way around, right, with the DNA on the top and the mRNA below. The bottom of this is the trip-L DNA. It gets transcribed. The ribosome starts translating. Okay, and if there's lots of trip, then the ribosome can get all the way along here to its stop codon. If there's lots of trip, it does not stall. It keeps going to its stop codon. And by doing that, it covers up region 2, preventing it from binding with 3. And instead, 3 binds with 4, and that makes a transcription terminator, and the polymerase falls off. So even though there's no repressor, even though the gate on the stream is open, we know that Dennis's house doesn't actually need water right now. Local control. We know that this mRNA does not actually need to be transcribed. And the other side of that, if Dennis does need a lot of water at his particular house, then the ribosome is going to stall on these trip codons. If there's not enough trip tRNA around, the ribosome will stall, 2 will pair with 3, and therefore 3 will not be able to pair with 4, and transcription will continue. So again, it's fine-tuned local control. What I want you to take away from this, aside from the mechanism, is this idea of layers of genetic control. Repressors, large-scale control, but attenuation is much finer. Attenuation changes things about 10-fold. The attenuator can drop expression down to 1/10. Repression, as we saw, could be 1/70th. So together we've got a 700-fold range of expression that we can work with using those two combined systems. Okay, terrific. You will find lots of animated versions of this on YouTube. Now that you have some background, have a look at that, and I'll see you again in a couple of days. Take care.